Yeah, thanks for giving me the chance here to speak this morning, this afternoon, this evening, depending on where you're at. The title of my talk is Prototyping Decentralized Science, and the goal here is to accelerate cures for rare genetic diseases. To start off, I just want to quickly tell you a little bit about myself. Professionally, I've been for 15 years in doing pharmaceutical technical development for a pharmaceutical company. And in my day job, I'm developing and manufacturing experimental medicines for clinical trials. In the field of DeSci, my experience level is much different. While I would like to describe myself as a very enthusiastic supporter for open innovation and collaboration, I'm very new to the DeSci community, even though the DeSci community is quite young. And today, a lot of what I'm going to talk about is a research project I'm calling DeSci Applied Research. Here, this is an independent research project to accelerate the advancement of genetic medicines for disenfranchised patients with rare diseases. I'll start out with a quick session overview. I want to take some time to introduce you all to some emerging technologies in the field of genetic medicine, present about some of the barriers to developing genetic medicines within our existing pharma industrial ecosystem, and then try to have a discussion about the applications of DeSci tools and principles to help overcome these barriers. We may not be able to do that online, but I will provide you some contact links so we can take care of this offline for people who are interested. All right, to get started, why do I care about this problem? The truth is it's a really big problem. So some quick facts about rare and genetic diseases. To start out with, there are estimated to be more than 10,000 recognized and means somehow diagnosed or published rare diseases and genetic disorders. And altogether, these can affect up to an estimate of 400 million people worldwide. Another interesting thing is about 80% of rare diseases are genetic in origin, meaning that this comes down to the fundamental biology of the patients, and about 50% of these diseases present themselves in childhood. And so what this means is that there are about 8,000 classified rare genetic diseases, and in terms of a treatment option, 95% of them have no treatment option. Now, a lot of the recent advances in biology and engineering are starting to change this in terms of what is possible, and from the science that, recent science and technology investments. The first technology I want to highlight here is next generation sequencing. This is a way of being able to go down to chromosome level understanding of DNA of an individual, and through this next generation sequencing, we're able to identify more and smaller patient groups that share common genetic anomalies that are contributing to the diseases that they have. One of the good news is there's also new technologies about how to fix some of these genetic anomalies. And so there's several of them that are under development. One of the most popular ones that gets talked about a lot is called CRISPR. And this is a suite of tools that are under research for biological applications. And I think actually the first CRISPR application for human use was just recently approved by I think it was the UK regulatory agency. But the important thing to remember is that each of these 8,000 plus genetic rare diseases is going to require a different fix because they're all caused by a different genetic anomaly. And then the last piece of technology that I wanted to highlight are engineered vectors for delivery. And so it's one thing to be able to identify that there is a genetic problem causing a disease. There's another thing to be able to fix that kind of in theory or in a petri dish. It's a very different and more difficult thing in order to deliver that genetic fix to the diseased tissue within the patient in order for them to have the therapeutic benefits. And so recent vector technologies based on different viral particles or lipid nanoparticles are now providing safer, cheaper, and more routine ways of delivering gene therapies to the patient. And so when I start to think about like the intersection between genetic medicine and decentralized science and what we talk about of being able to have more open use of intellectual property, and more collaborative ways of developing medicine. I think about the intellectual properties in two different ways. One, which I'm kind of showing on the left, these are non-fungible intellectual properties. Things like, what is the particular genetic fix for a particular disease that has been diagnosed? Um, and so these would be the genetic payloads, or like the CRISPR technologies that we would deliver to the patient. These are going to be specific to every unique genetic disease. For each of these technologies, you're going to have a limited and quantifiable patient population based on how many patients have this genetic anomaly, and that the value of this intellectual property or the value of this genetic fix goes up or is increased through the demonstration that it actually works in treating the patient in clinical trials. On the other side, there's other types of intellectual property that maybe is more open to be sharing, or I would call also as fungible. These are things related to a delivery and manufacturing process, and these are things that can be reused for many, many different types of genetic diseases. And so because it's very difficult, it takes a lot of time in order to develop one drug for one genetic disease, and that these can be used for many genetic diseases, you can think about it as a really practically limitless patient population because of the time and the effort it would take in order to reach all the patients who would be able to benefit from a particular delivery vector that reaches the heart or the liver or the brain or something like that. And the value of a lot of these, what I would call fungible intellectual properties, gains or goes up every time it's used. So in any time you would develop a drug and you want to test it in patients, there's always a certain level of risk. And by using this drug and demonstrating that it's safe to be used in patients, and that actually is delivering the genetic fix that we want without causing undue problems, or in the case of a manufacturing process, that is robust, that it doesn't cause you to lose a manufacturing batch or to somehow have productivity issues. This de-risking or risk mitigation from the use of this fungible intellectual properties kind of increases the value that it would have for the next researcher who wants to use it on their drug, because some of that risk that they would incur has been mitigated by the previous person who's been using it. And so now that we have some of these new these new delivery vectors and these new genetic medicines that are being developed, an emerging market is really forming, and it's really the emerging market is patients with rare genetic diseases that are somehow below the threshold in which pharma companies are financially able to support. And so what's happening is big pharma is advancing a lot of these tools that I talked about, while they're focusing on high prevalence indications, meaning diseases that are impacted by a lot of people in the population. So because big pharma is interested in these large disease areas, a lot of the technology and infrastructure is now being built in order to support the smaller patient populations. But the economic sustainability of supporting a small patient population where maybe you put all of the money into developing a drug, but you can only treat 50 to maybe 500 patients on the other end, it's hard to justify the low ROI when there's a larger patient population that in respects as well probably has a larger impact on the overall health of the society. And so patients, but it's not that people are ignoring completely the patients with rare genetic diseases. There's a lot of patient-led groups as well as researchers who are working on these rare diseases. And then at a lot of the industry conferences that I attend, as well as a lot of the journals that I read, there are calls for alternative business models to move these medicines forward. Models that are somehow more efficient at bringing niche products for smaller customer bases through the phases of clinical trials and then making them available to the public. And so for a lot of reasons, I think this niche market is an obvious entry point for DeSci into some mainstream pharmaceutical development. We've talked about how there's thousands of different diseases that are non-competing with each other because they affect different patient groups. We've talked about how there are technologies that are being developed, such as CRISPR, that can be customized to the different patient groups that have the different genetic anomalies. And then we've also talked about intellectual property such as delivery vectors and manufacturing processes or parts of manufacturing processes that can be used over and over again non-competitively to support multiple different drugs being developed for different patient groups non-competitively all around the world. But if DeSci is going to make a wedge into this part of pharmaceutical development, it's going to have to be by solving a problem that traditional business cases or traditional business models are not able to solve. And largely this is that the economics for rare disease drug development is really unsustainable. Already you have the problem that small patient populations, they're not going to get any bigger. Of course, we don't want them to get bigger. And so any cost that goes in developing a drug has to be burdened onto a smaller number of people. A lot of genetic medicine, because it is very new in terms of how long the technology has been around, as well as the complexity of delivering a gene therapy over a small molecule or even a biologic, means that the price already is unaffordable in the United States and across Europe. Therapies that follow some of these modalities are have an asking price of $2 to $3 million per patient, which for a very small number of patients, healthy economies or strong economies in countries like the United States and Germany and Switzerland and France, they can afford this, but they're already bulking at it. And then it also does limit access to patients who are not fortunate enough to live in those countries, but may still have the disease, but would still benefit from these drugs if it would become accessible to them. So what DeSci needs to be able to do in order to not impact negatively the price and the small patient populations is needs to be able to address something around the high development costs, and as well as the significant upfront risk in terms of how you're developing a drug where in the over however many years we've been monitoring drugs that have been approved by the FDA or the EMA, maybe 1 in 10 drugs that enters clinical trials actually is approved. Actually, I think I have more statistics about that on this slide. So when I talk about the process of developing a drug, and I talk about using decentralized science in order to foster collaboration and to solve the problems of drug development for rare diseases, what I'm trying to focus on is this area that I'm showing here in orange and yellow. So what I'm showing on this slide is kind of a really rough sketch of the drug development life cycle, starting from discovery and lead optimization, where there might be hundreds or thousands of different drug candidates that are being researched at different universities, different hospitals, or even small companies and foundations, largely funded by government. And this takes some time, and out of these hundreds of thousands of them, maybe 10 to 20 of them are shown to be successful in terms of in vitro studies or in mice. And these enter what is called a preclinical phase. And this is the generation of the data that regulatory bodies want to see to show that this drug is going to be safe and potentially even effective enough that it's worth the risk to try it in patients. Of the 10 to 20 drug candidates that enter this phase, 6 of them end up actually making it into clinical trials where we get to test, where they are tested in patients. And in the terms of gene therapies for rare genetic diseases, often in these phase 1 trials, they are patients who are actually presenting with this disease and could benefit from the experimental compound if it's one that works. And then what happens is out of the 6 or so that enter phase 1 trial, maybe 1 of them is successful there and goes into phase 2 versus commercialization. And then in terms of becoming a commercial product, it's at this point where pharma mergers or acquisitions may be in licensing the technology in order to carry it the rest of the way. But what ends up happening for a lot of these drugs being developed for rare diseases is they get stuck somewhere in here between preclinical and clinical phase 1, because the costs in order to do this development goes up almost exponentially as you go from having to do very quick and dirty experiments in order to treat or try to treat like a mouse model, to having a much higher quantity as well as quality material in order to get regulatory approval, but also like as a researcher, the confidence that what you're putting into patients is is going to be safe, as safe as we can ensure, as well as give them the best possible chance that it's actually efficacious and is going to do something for them. So you have a lot of drugs that are going through these phases, and then throughout the pharma industry, there's probably hundreds, of siloed research efforts for different gene therapies that are going on at any one time. And remember what I started out with: there's 8,000 plus non-competing genetic diseases or indications. Here's another word that we can use for a disease or disorder that could benefit from this technology. And so what I'm trying to show, or what I'm trying to explain to you, is that in terms of decentralized science and collaboration between different organizations, you have a lot of people who are struggling with the same problems while trying to develop the same drugs or similar drugs for very different patient groups. And so it's not like they're competing with each other, but they could be collaborating with each other. But in the traditional way of doing this sort of research, it just doesn't happen. So if we think about DeSci as a solution to how to develop drugs for indications that large pharma is not very good at, it doesn't have to solve these problems in a vacuum. And so there is a very strong functional ecosystem for developing drugs that can be adapted to DeSci principles. So what I'm showing on this slide across the x-axis here in the swim lane diagram is a lot of the same phases that I showed in the previous one, where drug development starts with fundamental research and early discovery. There are the phases of preclinical development and clinical trials, and then drugs that are successful in clinical trials, they can go and be launched as new products. And then in the ecosystem of companies and foundations and institutions, and most importantly, the researchers and the scientists and the patients, there's a lot of different kind of categories of players who are participating. And they're not all the large pharma companies and the venture capital companies that somehow become the villain of the industry. And so when we think about how a new gene therapy for a genetic disease is kind of flowing through this development life cycle, you have to also realize that it's already a collaborative effort between lots of different organizations, often starting at the very beginning with some research center. Maybe some of you are part of these research centers at different universities or working at hospitals. And often these groups are pairing up with rare disease patient groups in order to understand the disease, in developing the motivation in order to try to come up with a cure. And then as we move to the right and we move to the later phases of the life cycle, there's different organizations that get involved in order to complete different activities and different specialties that need to happen. Something like a translational institute will do a lot of the drug optimization, as well as doing a lot of the adaptation of manufacturing platforms and producing IND enabling studies, which is basically saying the studies that you need to complete in order to show to a regulatory body to say, hey, I'm ready to test this product in patients. There's some very clear guidelines on what is needed there. CMO stands for contract manufacturers, and CDMO stands for contract development and manufacturers. These are companies that they have a lot of scientists and engineers, as well as technicians who know how to do wet lab work and manufacturing. And these are organizations that you can contract out to hire in order to do a lot of the heavy lifting in order to make enough of your experimental drug in order to put into patients. Contract research organizations, these also, again, contractors for hire. They're less focused on the chemistry and the manufacturing and the biology, and they're more focused on the design of clinical trials and what are the number of patients that you need, how do you need to measure the clinical efficacy of these drugs in these patients, basically saying if you give a patient a placebo versus the drug, what are you looking for in the experimental arm of that study that you would write into a protocol that says, if I see this, then I know that this drug is working. And they oftentimes have to work with the research center or the patient groups in order to try to come up with clinically meaningful endpoints or clinically meaningful outcomes that would translate into this drug being efficacious enough that the patients would actually want to take it because they think it does something that somehow is improving their lives. And then if all of these people are successful in the rare disease indications, sometimes a pharma company will come in and acquire those assets, do further clinical trials, and then try to commercialize it. Other times, if it's necessary, a venture capital company may come in and do the funding. But often for very rare diseases, and because there's so many rare diseases, the attention of the few pharma companies and venture capital companies who can invest in here is limited. And so if we want to map on some of the most challenging parts of this life cycle, this development life cycle, where a lot of rare disease therapies get stuck, and also things where having more improved and more efficient collaborative tools could be helpful. Here I've highlighted now in orange in terms of the fundamental research. Even doing something like a natural history study, which is finding patients of rare diseases that might be all over the country or all over the world, and trying to track how does that disease progress, the same disease in all of these different patients, to try to get a distribution of like what you would expect to see in a placebo arm of a clinical study, and so that when you do preclinical development and you try to design a trial, what are the endpoints or what are the measures that you would expect to see in an experimental arm relative to the natural history of the patients that tells you that this drug is actually working. When it comes time to do clinical trials, like I said, if the patient numbers are small and they're spread all over the country or all over the world, recruiting those patients and getting them into treatment centers and so that they can be administered with either the drug or the placebo, that creates an additional challenge. And then as we want to do developments and manufacturing of these drugs, access to the key intellectual properties that I talked about, things like how to operate a CRISPR gene therapy, how to deliver this therapy using some sort of vector, and then also how to manufacture it in a way that it's affordable. These are all things that get sort of tied up behind patents and behind or in silos of companies who can afford the intellectual property, and then they're sort of withheld from the development of drugs that are for patient groups that are not of interest of that holding institution. The second to last challenge that I wanted to talk about is supply of skilled workers. So, like I said in the beginning of the talk, a lot of this, being able to treat rare diseases with gene therapies, it's an emerging market. And over the last, maybe 5-10 years, it's actually grown quite fast. And so what has happened is that you have a lot more of these institutions, translational institutes, CMOs, CDMOs, popping up, all over the world, which is actually really great, right? Because now you have a lot of other places you can go in order to, acquire these services to develop your drug. The downside of that is that our universities and our educational systems are not kind of producing enough skilled workers in order to keep all of these different places fully employed with people who have the knowledge and the background and the hands-on skills in order to develop these products. And so kind of enriching the skills of laborers who are in here working in this space, as well as encouraging people who are thinking about, what do I maybe want to do for my career, and if, how can I get into science and get into cutting edge science and really make a difference in patients' lives. We need skilled workers like that all over the world within these companies in order to do the work. And then the last point I have here on the bottom is funding. And so I think this is something that anybody who has worked in research, especially if it's publicly funded research or research onto early stage projects or more experimental projects, funding is a difficult topic, and gaining funding is very complicated sometimes. So my initial plan was to finish the talk here and to have a discussion, which I don't know if is going to be possible. It doesn't seem like it was possible for the previous speakers. But I will have my contact information in the end, and so I'm happy to continue this conversation offline. If we were to have a discussion, I openly would want to ask the questions to the audience, people who are more experienced in decentralized science, and the tools and the principles. Can DeSci be mapped onto a functioning research and development ecosystem? So I think I showed in the swim lane diagrams, there's already a lot of players out there. It's a growing industry. It's an industry that has a lot of skills, and a lot of strong background in different areas that are not necessarily broken. They're just not working very well together. And so can DeSci be something that improves collaboration amongst the different people or the different research organizations working for different patient groups and trying to navigate through this environment in a silo? And if the answer is yes, the question that I really wanted to get to is, where can decentralized science solve prevalent problems in the development of rare diseases? And so some of them I highlighted here were insufficient funding that cover the high costs in development. Another is the high risk exposure to each individual project, the diluted workforce in the growing industry sector, and then finally the design and execution of decentralized clinical trials and natural history studies. And so if you want to engage in this conversation, and who knows, maybe we can have a follow-up virtual meeting where we can hold these discussions. Feel free to reach out to me. My email address is chris@DeSciAppliedResearch.com, and on Telegram and in the BanklessDeSci or group chat, I'm PharmaTokens Chris.